people who delight in controversy should enjoy studying the life cycle of the slime molds or Myxomycetes. The generalized life cycle usually given in textbooks is as follows. A spore germinates, giving rise to a myxamoeba or swarm cell. A myxamoeba may develop flagella and become a swarm cell. A swarm cell may lose its flagella and change back into a myxamoeba. Under unfavorable conditions, a myxamoeba or swarm cell may encyst. When conditions again become favorable, the cyst may germinate, giving rise to a myxamoeba or swarm cell. A myxamoeba may divide. Eventually, the myxamoeba or swarm cells fuse in pairs, forming zygotes. The zygotes may grow by themselves, or fuse with other zygotes and develop into a multinucleate mass of protoplasm called the plasmodium. Under unfavorable conditions, the plasmodium may form a resting body called a sclerotium. The sclerotium is composed of macrocysts. Under favorable conditions, the sclerotium may again become a plasmodium. Eventually, the plasmodium converts into one or more fruiting bodies which bear the spores. The spores, myxamoebae, and swarm cells are thought to be haploid. That is, they contain only one set of chromosomes. The zygote and plasmodium are thought to be diploid. That is, each nucleus contains two sets of chromosomes. The change from haploid to diploid is the result of fusion between myxamoebae or swarm cells. The change from diploid to haploid is the result of meiosis and is thought to occur during the formation of the fruiting bodies. Where is the controversy? Different scientists working on the life cycle of different species have reported variations in the different stages and even in the overall life cycle pattern. Let us see how the life cycle of Physarum gyrosum fits the accepted pattern. Here is a germinating spore. Throughout this film, two numbers are placed at the bottom of the scene. The one at the left refers to the magnification at the film plane. The one at the right is the number of times the action has been speeded up. Let us take another look at spore germination. Notice that the spore wall splits and the protoplast crawls out. After putting out numerous pseudopodia, the protoplast forms a flagellum and swims off. In this species, the flagellum is formed immediately following germination. Sometimes the flagellum is formed while the protoplast is still in the spore or while it is crawling out of the spore. Sometimes the flagellum is not formed for several hours. Under certain conditions, it may not be formed at all. Here is a Fuligocinerea spore, which has started to split open. Within the spore is one nucleus, seen as a light area. This area becomes indistinct as it divides. The single protoplast then divides and two protoplasts emerge from the spore. This phenomenon is quite common in some species, but not in others. Here is a Stemonitis flavogenita spore, which is germinating. In this species, instead of the spore splitting, a pore is dissolved in the spore wall. This is common in some species, but not in others. If we look at a drawing of a swarm cell, we see that there are very few specialized parts. Yet this cell carries out important physiological functions like other organisms. At the anterior end is a light-appearing area, the nucleus. Within the nucleus is a dark body, the nucleolus. At the extreme anterior tip of the cell is a long whip-like flagellum. A second flagellum is often present. This is usually short and bent back against the cell surface so that it is difficult to see. At the base of the flagellum, inside the cell, are two small granules, the basal bodies. At the posterior end is another clear area, often in a loop-like appendage. This is the contractile vacuole. Scattered throughout the cytoplasm are small dark granules and irregular rods. These are thought to be mitochondria. Sometimes bacteria and other particles are visible in the clear food vacuoles. Here's a swarm cell creeping over the substratum. Many of the parts shown in the drawing can be seen in this cell. The nucleus, which is sometimes called the control center of the cell. The nucleolus, which many scientists think is involved in protein and RNA synthesis. One flagellum, which enables the cell to swim. The contractile vacuole, which probably regulates the osmotic pressure of the cell by bailing out excess water. And the mitochondria, which are sometimes called the powerhouses of the cell since they are thought to be involved in respiration, thus providing energy for cell activities. Sometimes very fine pseudopodia originate at the anterior end of a creeping swarm cell and migrate over the cell surface to the posterior end. In this swarm cell of Stemonitis flavogenita, the pseudopodia resemble flagella. This raises the question, how many flagella does a swarm cell have? 1 or 2? Most scientists think that the swarm cells have 2 flagella in addition to the migrating pseudopodia, as shown in this Fuligocinerea swarm cell. What function do these pseudopodia have? We do not know. Perhaps they aid in locomotion. Perhaps they aid in feeding. Here is a Didymiomeirides swarm cell, which shows one method of feeding, the engulfment of particles by pseudopodia at the posterior end. Another method of feeding usually attributed to the swarm cells is by the uptake of nutrients in solution from the surrounding medium. In all of the preceding scenes, you have seen creeping swarm cells which have a flexible shape. Here we see a swimming swarm cell. This cell is shaped like a comma or corkscrew. It swims in a spiral. Its long flagellum is directed anteriorly. One might expect the flagellum to push the cell backwards, but it does not. The flagellum undulates in a spiral and is directed at an angle so that it causes the cell to rotate and gyrate. The cell pulls itself along like an airplane propeller. Here's a swarm cell which is resorbing its flagellum and changing into a myxamoeba. The myxamoeba has the same structure as the swarm cell, except that it has no flagella and is irregular in shape. In these dying myxamoebae, we can see the internal parts clearly. The nuclei are centrally located in the cells. A cluster of mitochondria are at the bottom of the cell at the left. There are definite membranes around the mitochondria and nuclei. These myxamoebae are shown at normal speed. Notice how slowly they move. Here the action is speeded up. The myxamoebae are engulfing fungus spores and other small particles. All undigested particles are left behind. The swarm cells and myxamoebae of different species vary slightly in size and shape. The nucleus in a myxamoeba divides by mitosis as in other organisms. It is thought that the chromosomes reduplicate themselves and split. The 2 groups of daughter chromosomes separate and move to opposite poles. The cell cleaves by a pinching in through the equatorial plane. The division process is shown here by animation. During prophase, the chromosomes become visible as minute rods. The nucleolus and the nuclear membrane gradually disappear. The chromosomes move to the center of the nucleus and align on the equatorial plane during metaphase. A spindle is formed. During anaphase, the daughter chromosomes separate and move to different poles. Following telophase, the nuclear membrane and nucleolus reform and the chromosomes become less distinct. The cell then divides into two equal parts. Let us take another look at cell division under the microscope. The cell has rounded up. The nucleus is in prophase. They have now aligned on the equatorial plane. The nucleus is in metaphase. The two groups of daughter chromosomes separate during anaphase. The nuclei are in telophase. The cell begins to cleave by the pinching in process. By speeding up the action, we can follow the reformation of the daughter nuclei and the separation of the daughter cells. Encystment is a gradual process. Here, a myxamoeba rounds up. It gradually becomes reduced in size and forms a cyst wall. The mitochondria occupy a position around the centrally located nucleus. This cyst may germinate later when conditions become favorable. Fusion may occur between two swarm cells. These cells come together at their posterior ends, which are usually sticky. The fusing cells swim erratically since the flagella of each cell beat in opposite directions. The fusion process may last as long as several hours. Eventually, one mass of protoplasm is formed. Myxamoebae may come in contact one with another without fusing. Then, without any forewarning, the cell membranes break down and the cells rapidly fuse. This fusion of protoplasts is followed by a fusion of the nuclei. Nuclear fusion is gradual. The nuclei adhere to one another. Gradually, the nuclear membranes break down and the two nuclei become one. Usually, their nucleoli also fuse. The resulting cell is a zygote. A zygote is usually larger than a myxamoeba. Its nucleus is also usually larger and its cytoplasm is often more granular. The nucleus may undergo mitosis as in the myxamoeba. Mitosis in the zygote is shown here by animation. The process is similar to that found in the myxamoeba, except that the equatorial plane is larger, there are more chromosomes, and the nuclear membrane persists at least until telophase. The protoplast does not divide following mitosis. Let us watch mitosis in a zygote as seen under the phase microscope. The nuclei are in early metaphase. The two groups of daughter chromosomes separate and move to opposite poles. The spindle appears to stretch and constrict in the middle. The two daughter nuclei become indistinct. The protoplasm does not divide. As we speed up the action, the nuclei become visible and we see the nucleoli forming from fusing granules. The result of mitosis in the zygote is a two-nucleate plasmodium. A young plasmodium is shown here at normal speed. The cytoplasmic streaming is slow and irregular. If the action is speeded up, we see that the plasmodium moves about and engulfs particles. Sometimes a plasmodium engulfs a myxamoeba and digests it. After a period of over 12 hours, the nuclei in a two-nucleate plasmodium divide by mitosis. The divisions are similar to that in the zygote. Both nuclei divide at the same time, resulting in a four-nucleate plasmodium. These synchronous divisions may continue at regular intervals. Here is a culture of Physarum gyrosum at a low magnification with the action speeded up nearly 3,000 times. The small light dots are bacteria, myxamoebae, and swarm cells. The larger light areas are zygotes and small plasmodia which grow and fuse. Some workers claim that in certain species, a plasmodium develops from a single zygote, whereas in other species it develops from the fusion of several zygotes. As seen here, large plasmodia may engulf myxamoeba or fuse with zygotes and other plasmodia. In Physarum gyrosum, 3-nucleate plasmodia, such as this one, result from the fusion of a zygote and a 2-nucleate plasmodium. Fusion between plasmodia is common. Here is fusion between a 3-nucleate and a 4-nucleate plasmodium. Note how quickly the fusion occurs. Here is fusion in fuligocinerea between a 2-nucleate and a 4-nucleate plasmodium. As a plasmodium gets larger, certain changes occur. Pigment granules develop. These are seen as highly refractive granules. The cytoplasmic streaming becomes confined to channels and becomes reversible, as shown here. A plasmodium gradually changes from round or amoeba-like to the net-like shape of the mature plasmodium shown here. An actively moving plasmodium has a fan-like anterior end with many trailing veins. When food is plentiful, the plasmodium usually thickens and remains immobile. If we look at several of the veins under higher magnification, we can see the reversible cytoplasmic streaming. Not all species have a plasmodium resembling this one. In the few species of minute slime molds which have been investigated, the plasmodia remain small and amoeboid. Streaming is irregular, as in the young plasmodia of Physarum gyrosum. Taking a look at a plasmodium under an even higher magnification, we see that the nuclei, mitochondria, pigment granules and vacuoles are carried along in the stream. The plasmodium of Stemonitis flavogenita is nearly invisible to the naked eye. If we examine it under the microscope, we see fine veins which resemble fungus hyphae. Using still higher magnification, we see the characteristic streaming that we saw in Physarum gyrosum. Notice that the thick outer gel layer is limited or lacking in Stemonitis, and that the cytoplasm possesses large vacuoles. A Stemonitis flavogenita plasmodium will form macrocysts if the environmental conditions become unfavorable. A Physarum gyrosum plasmodium will also form these macrocysts. In this and similar species, the cysts may form a hard body called a sclerotium. The sclerotium is taken and is reforming a plasmodium. When conditions are just right, the plasmodium thickens and clumps. In Diderma testatium, these clumps of protoplasm become the fruiting bodies. These fruiting bodies are arranged in the shape of the plasmodium. As the fruiting bodies dry out, lime becomes visible on the surface. In Physarum gyrosum, the plasmodium thickens, clumps, and forms papillae. These papillae fuse, constrict, invaginate, and twist to form an irregular gnarled mass. The fruiting body changes in color from cream to black as the nuclei divide and the spores are formed. Here, Trichia and Stemonitis are fruiting at the same time. Taking a closer look at fruiting in Stemonitis fusca, we see that black stalks are formed inside the white papillae. The protoplasm moves up the stalks as they are formed. The color changes as it did in Physarum gyrosum. In Stemonitis fusca, the outer membrane covering the fruiting body disappears as the spores expand and dry out. In Cerathiomyxa, the spores appear to be formed externally on the fruiting body, although not all investigators agree as to how this process occurs. In Dictydiathalium plumbium, many papillae fuse to form one large fruiting body. As you have seen, the fruiting process varies in different species. The reduction of chromosomes is thought to occur during fruiting. This reduction process is meiosis. Here are scenes of stained slides made from developing fruiting bodies of Stemonitis fusca. The cytoplasm cleaves into cellular units as the nuclear divisions begin. All the nuclei undergo two successive divisions at about the same time. This is meiosis. The cells divide two more times, resulting in uninucleate spores. Sometimes these events do not occur in this order. Scientists differ in their interpretation of these events in different species. As you can see, it might be possible that several nuclei would end up in one spore, complicating the next life cycle. You have seen a few of the critical events in the life cycle of Physarum gyrosum and of several other species. The life cycle of Physarum gyrosum resembles the generalized life cycle described in textbooks. This consists of spore germination, myxamoebae, swarm cells, fusion, zygote, plasmodium, fruiting bodies, meiosis, and spores. Do all species follow this pattern? What are the variations in the different stages, and what causes these variations? We do not know the answers to these questions and must therefore continue to study the controversial life cycle of the slime mold.